Okay, so this is part 2 of our discussion about DNA replication. And in the first part, we talked about how we figured out that it was semi-conservative replication. And now what we want to do is we want to move on to discussing the major players that are going to come in and basically the mechanics of this. And what I suggest to everyone is there's some nice YouTube videos. There's other videos out there. You might want to watch those. Some are accurate, some are less accurate, but they give you the picture. And in this part, what you really want to do is you kind of want to think of it yourself as a movie, right? There are steps that we're going to talk about. Things come in, things go out. If you can think of it as a movie, and you can stop it at any place, and then, as you're studying, say, okay, I'm stopping in here. What just happened? What just came in? What just left? What am I looking at? That's a good way to study for this part, okay? Because there's a lot of complicated little parts that we're going to throw in. Now, most of what we know about replication started with Arthur Kornberg back in the 1950s. He won a Nobel Prize for his taking these ideas of Watson and Crick and others and putting some information to it. Now, unfortunately, really what we know about replication for the most part comes from studying bacteria, not human DNA and others. Now, we're learning more and more and more constantly, but really from prokaryotic systems is where we know most of what we know. And so we know some additional new things with the eukaryote, and that's building by the day. But what I'm going to do here is I'm going to talk about these things and the names that are used, and a lot of the ideas are really, and this is consistent with the textbook, are really from bacterial systems. But the same basic steps have to happen, and do in fact happen, whether you're talking about human DNA or whether you're talking about a bacterial DNA. Okay, so let's start out. We've got this idea that we've got this double-stranded DNA. We're somehow going to have to break it apart, and we're going to copy each side. So we have an original strand, and then we have a new strand. Okay, so we're going to work from that idea. We're going to break it down into 4 big steps, just so we have some way to think through it. We're going to talk about the initiation process, how you get your DNA then unwound once you initiate the process of replication. Then we're going to talk about primer synthesis and how we lay down and get started, the start process, and then we'll talk about elongation, how you actually do the process. And then at the end, we're going to talk about how we fill things in, but that'll be in the next lecture. All right, so initiation. We'll start here. All right, first off, there has to be things called origins. Origins of replications are places where replication starts. You have circular DNA, like in the mitochondria, you'll have usually one origin point. If you have a long chromosome, like you have in Drosophila or in humans, you'll have many origin points. Okay? These origin points are where you have replication start, okay? And they start there by having things called initiator proteins. These are proteins that come in, bind to the DNA, and they set up the process of starting the whole operation. Once you have an origin start to open up, you get what's known as a replication bubble. The bubbles get bigger and bigger. Replication goes out in what's known as a bidirectional pattern, and you end up finally with replication forks combining each other. In human systems, we have between 30 and 50,000 origin points. So we've got 23 sets of chromosomes, right? So we've got 50,000 or so points spread around. Others, fruit flies and things, have maybe 10,000, 12,000. It just depends on the organism. The point is, there are lots of different origin points. You start here, you start here, you start here, you start here, almost all simultaneously done. Okay? Once the origin starts, you get these things called replication bubbles, replication eyes that you can see. Those are places where you've separated the double-stranded DNA and you started putting in the new strands, shown here in blue, and you're working bidirectionally so they move together. These bubbles will get larger and larger and larger, and eventually they will run into each other because you're going in both directions. When they run into each other, they fuse the DNA, the new DNA that you're making. They can connect it all up so that at some point you actually have an old strand, a complete new strand, a new strand, a complete old strand. If semi-conservative replication is completed. Okay? This is a picture of showing the actual replication eyes that are opening up. These strands that you see here are double-stranded DNA. They've opened up, and you've already started the process of copying. So this would be a replication fork right here. This would be a replication fork right here. We'd be working this direction, and we'd also be working this direction. You can see 2 have opened up here, and they've almost connected with each other. So once they connect, they'll fuse that DNA together, and you'll have just one continuous strand. Same thing going on here. This one's obviously been going on just slightly longer than this one, but this process, this will go this way, and eventually this will move this way, and those will all interconnect. All right, now how does this happen? Again, we're going to talk primarily about bacteria as far as the naming of these things, but there has to be similar processes going on in eukaryotic, and we know that there are. The origin of replication, these origin points are usually AT rich, meaning they have lots of A's and T's. That's how you find it. Okay. There are these things called DNA A boxes, which a DNA protein comes and adheres in these areas near these A and T's. Now, DNA boxes are again described for bacteria, but essentially there's a complementary type of protein complex that forms as well as everything else. And also, we'll talk later about methylation, but there's gamma methylation sites that occur along here too. DNA can get methylated, and these methylation sites are places where you can open up the DNA and start the process. Okay. Now, all this occurs by what are known as initiator proteins. These are origin-binding proteins. So here's your chromosome. Some spot on it has the right code for it to be an origin, and here's this complex of proteins that come down to work there. Now, this is not going to make you try to memorize or learn this particular figure. It's too complicated. What I'm basically trying to show you here is that, in fact, it is a fairly complicated system. Okay? Here's your double-stranded DNA up here. Here's some code within that DNA to say this is one of your many thousand origin points. You have to have an origin complex come in. That complex pulls in other proteins. MCM proteins are one of those that will come in, and other MCM proteins will come in. This is your pre-replication complex. This is before anything happens. These are just locking down on here. A whole series of other elements come in, mostly proteins, but there's some RNAs and other things. And then eventually DNA pol, we call it pol for polymerase, will come in. Once that comes in, you initiate the complex. There's also this germinin protein that comes in and stays with the system until the process is over. Okay, so the point of this is you have an origin. The origin is, once you start the replication, the origin is recognized by this complex that says this is an origin point. That draws in lots of other players into the system, and now once you get them all in place, you're ready to start the actual process of breaking these little hydrogen bonds and starting to replicate. Okay. Now, how do you know when you hit an origin point that anything should happen? Okay. Well, first there's this what's known as an origin replication recognition complex, the ORCs. These remain during the DNA during the whole process. So they come in and they sit there. Okay. Then there are these other proteins called lysosine factors. All right. What you want to do is when you go into replication, so you're in G1 of the cell cycle, you go into replication, you want to say, okay, these spots need to be replicated, and you start to do that. You don't want to keep doing it. You don't want to go back to the same spot 5 times and try to replicate DNA. You'd have a mess. So what we have are things called lysosine factors. And lysosine factors and the process of licensing is simply saying this is a place, an origin that you can go to, and you can start the process, but don't go back to that origin. Okay? And this is basically what happens is that you have the CDC6s and the CDT1 proteins. They bind, they coat the DNA, and they start the process. Again, you're not going to have to learn all these. You need to learn what licensing is as a general thing. You need to learn what origin recognition complex is and well, okay? Now, once you go into synthesis phase, these proteins disappear. They leave, okay? And you have this front of these other proteins, these MCM proteins that are these little bubbles right here, serve as the advancing replication fork. So they're sort of out there helping the process move along, okay? Now, just to take a little side trip for a moment, there are a number of genetic issues, genetic problems that we've starting to discover that associate with these things. The Meyer-Gorlin syndrome is a syndrome where during fetal development, the development phase slows down. Bones and other things don't grow as fast. There's also microcephaly, with the head small. These are individuals essentially that just don't grow very quickly during the normal phase that you would during embryogenesis. So during gestation, they sort of slow. One of the reasons they thought that possibly happening is that there is a recessive mutation that occurs up here in the CDT1 gene, and it's back in the gene, right, that makes this a dysfunctional protein. And in doing so, it slows down the process. It doesn't come in when it should, or there's not enough of the proper protein, and so these individuals simply aren't signaled to go through meiosis as they should, so therefore they don't develop as quickly as they should. There's other mutations that are known to be associated with this, and there's a lot of ambiguity as to whether this is the actual cause, but it certainly has something to do. We know this mutation is associated with this syndrome, and it's likely that it's at least plays some role, either directly in replication or in some secondary role of the protein. Okay? Now, we have this positive control. Positive meaning that you have to have the licensing factor there. It's like putting the key in the car, right? Won't turn over to your key or whatever your fob is near it. Then there's a negative control as well. You saw back before that the germline came in. It's a protein that once you go through the replication process, okay, the G2 up here, you start to add this protein, okay? And what it does is it basically stops this process of synthesis. It blocks the action of some of these other proteins and slows it all down. Once you go through mitosis, as part of the process of mitosis, it starts to degrade this protein. So by the time that you enter G1, the germline is basically gone. You can start rebuilding the licensing factors and preparing yourself to go back through S phase. Now, if it's not going to go through S phase, of course, then it phases out into G0. But if it's going to replicate again, then you start the whole process over. You start building these accessory proteins, licensing factors, getting them all ready, cleaning up the origin points so that they're ready to be reused. Okay, so this is how it works. You've got both things. You've got licensing, which says yes, you got to go here. You get rid of the licensing, you can't go back. But you also, as a secondary protection, have this other protein that also prevents this buildup. So even if you somehow got in there and started to pull those things apart, you would stop that process as well. Okay. All right. So we've initiated. We've got everything sitting there. We're ready to start pulling our DNA apart. That's what we're going to call unwinding process. Okay. Under unwinding, we're going to have three basic things come into play: helicase, single-stranded binding proteins, and a topoisomerase that we're going to give a special name. Okay, this topoisomerase is known as gyrase most often. Okay, it's a general name. And again, it's a complex of proteins. But what it does is that, you think about it, if you take a rope that's coiled up with a double-stranded or three-stranded rope, and you really pull the ends apart really quickly and try to separate that rope, it just makes a knot. Okay. Well, the same thing happens with your DNA. You start to pull those pieces apart, the DNA starts trying to flip around and makes a knot, right? Well, what these topoisomerases do, what gyrase does, is nick the DNA so that one side will spin around and the other side will stay in plane, and it releases some of that tension. Now, while that's going on, helicase is involved, and helicase is opening up, breaking these little hydrogen bonds. When those are broken apart, single-stranded binding proteins come along, and they stabilize these single strands. Because remember, DNA is really sticky; it likes to go back together, and so there's got to be some way to hold it apart long enough for the other processes to go on, right? So, helicase, that's breaking apart the strands, causing the replication fork. When that's going on, you have topoisomerases, or gyrase in our case, which we'll call it, downstream somewhere, that's helping you keep this supercoiling from happening, and you've got these proteins that are helping keep the DNA single-stranded. All right? Now, once we get it open, we've got to start the process. But it turns out that you can't just start putting DNA nucleotides on there. There has to be some building block that comes along. All right, and that building block, that protein, we're going to call primase. Now, it puts down an RNA primer. Okay, so this is just instead of putting DNA down to start with, you actually put down a short primer. This primer is somewhere between 10 and 12 bases long. So it'll come in the replication fork, it'll lay down a primer in a 5' toward 3' direction, and it'll just put this little RNA piece in here. Okay, and it's an RNA primer, and that's called primase. The primers sit there. They are the first place. At the end of that primer is where the first DNA nucleotide will be hooked on, and they keep going until they move further down. Okay. Eventually, another piece of DNA polymerase, the one that's producing the DNA, will come along like this, bump into that primer, and actually remove it, eat it away, and replace it with DNA. It's part of the process of moving on. So primates. Now, if you read the book and you look other places, primase is really a fancy form of DNA pol, of DNA polymerase of a certain type, and it's part of this whole complex. But we're trying to keep the pieces separate, so we're just going to call it primase for our purposes. Okay? All right, so now you've got a primer, you've got it open, now we've got to elongate it. We've got to actually make the molecule work itself. Okay? That's done with a series of enzymes known as DNA polymerase. DNA polymerase are a group of enzymes that are themselves have kind of multiple components, and they have a catalytic site. A catalytic site is a site where a protein folds up, and inside that site, activity occurs. In this particular case, what's going to happen is you're going to bring in NTPs, you're going to bring in deoxynucleotides, you're going to build phosphodiester bonds, and you're going to then allow these little links to occur between the bases of the complementary strands, these hydrogen bases. Okay? There's lots of poles, okay? And certainly when you start getting into eukaryotic systems, you'll see that there's all kinds of poles. There's pole 1, which removes the RNA primer. And often they're called alpha, beta, different names. We call them pole 1. Pole 3 is the more common one. That's really the one that does most of the action during replication. There's a pole 2, 4, 5, 6. I think there's like 13 different poles now. Some repair DNA later, some proofread. They do all sorts of different things. Okay, we're not too worried about that. Remember, this is genetics. We're trying to just understand the process here. Okay, and we're getting down to what we want to talk about. But right now, we're just saying, okay, piles in there. Once you take cell biology or molecular or biochemistry, you'll learn all these poles and all their activities. Okay, big points for us. These polymerases, whichever one they are, they can't by themselves just start putting down nucleotides. They need a primer. So they have to have an RNA primer, and then they link to that, and then they move through. Okay. Secondly, they can only work in a 5' to 3' direction. So here's the sense strand, or the strand that's the parent strand up here, the one you're copying. You can only lay down by putting a base here, then a base here, then a base here, and a base here, and working toward a 5' to 3' direction. The 3' end will always be hanging out here somewhere. Okay. So you can see this lightly colored one is the 5', the new strand is being made, always being built 5 to 3. Okay, so the elongation process looks something like this. We're going to bring in a triphosphated NTP, whatever it has to be. If there's a G over here, you'll bring in a C. There's an A, you of course bring in a triphosphated T, TTP, and then you form a phosphodiester bond by giving off some energy and have it. So you're going to have these nucleotides triphosphated. They're going to come in. You're going to build a bridge between using the 3' that's hanging off here, OH. You're going to cleave off of these phosphates. Are going to be for energy, right? And you're going to form this phosphodiester bond coming back. The next one will obviously come in right here, and the next one will be here, and the next one will be here, and it'll be complementary to whatever's on this original parent strand that's over here. Okay. All right. So, just to remind you, this process is semi-conservative and it's bidirectional. So we're going to open up the fork, we're going to put out a primer, and we're going to have one strand called a leading strand where we're just making a continuous addition of nucleotides. We're opening up the fork, helicase is in action, then we'll just keep moving, keep moving, keep moving, keep moving, keep moving. The other strand, because the DNA polymerase can only work going this way, right? It can only work in this direction. You can't do that. What you do is you lay down primers, you make a little short segment, you have another primer, you lay down a short segment, and you work. So there'll be, as soon as this opens up here, a primer will be put down right here, and you'll make a new piece of DNA down to this primer. This will go a little further. You put down another primer over here. That's called the lagging strand, because replication occurs simultaneously up here and consistently. Here it has to break up. So the slow process of breaking up plus removing these primers slows it down. So you have a leading strand and you have a strand that's a little slower, known as a lagging strand. All right. Synthesis occurs basically like I said. You come along, DNA polymerase, it moves along, you get the development of the phase, and from a lagging strand, you get primer, a short piece of DNA. In bacterial genetics and other things, these are called Okazaki fragments. That's fine if you want to call them that. They're little fragments, and you form, okay? Now, what has to happen is one of the very important things is when you knock off a primer, it doesn't matter if you're on the leading strand and you eventually run into a primer down here, or you're on the lagging strand and you run into one of the mini primers. As soon as DNA polymerase removes that primer, like it's doing here, there's one little gap that's left. And that one more phosphodiester bond has to be put right here. In order for that to happen, you have to have DNA ligase come in. It's another form of DNA pol. We're going to call it ligase. To ligate something means to join it together, okay? So this ligase comes in, it's a specialized DNA polymerase that's going to just bridge that one little gap right there between where the old primer was and the end of this new piece of DNA that's coming along. Again, it happens both on the leading strand and the lagging strand, but obviously it's much more common on the lagging strand. All right, DNA ligase joins together the newly synthesized strands to form this form, okay? All right, we also, of course, have to have things like proofreading, right? Okay, how do we work with proofreading? Well, it turns out that there's a mismatch if you say this is an A and you put a G down here, it recognizes that there's a problem there, and it will actually proofread. Now, the proofreading occurs in the middle of it, but again, this is a molecule that's moving this direction, right? It's moving toward the right side of the screen. It checks to make sure, and obviously, if you're linking up a G and an A, you're going to have a misconfiguration of what's going on. So it tends to knock the new strand. It tends to knock that piece off and replace it with the T that's supposed to be there. It's very, very effective, and it results in a high rate of fidelity. Okay, this is called proofreading, which is reading to see if it's correct, and it does it for every base pair. And then exonuclease activity means that it knocks the ones that are off. If they're wrong, it simply removes it and brings the correct one back in. So it's doing that constantly as the system moves along. Okay? All right. Now the last part of this is to explain how can you have sort of a simultaneous production synthesis if you've got one strand going one way and one going the other. Well, it turns out that this DNA polymerase complex actually grabs both strands. It grabs the leading strand and just moves along. The lagging strand isn't the separate part here. It actually forms a complex where you got almost a lariat. It's called a trombone loop, but it's basically this loop that's right here. So you have a loop to where the primer is going down, and this trombone loop comes along like this, where you have the DNA. So what's happening here is you're grabbing this and making this pieces as you go. It's one big complex of form. Here's probably a better picture of what's really going on. Okay, we have the primer sitting down here. We have the lagging strand sitting here, and this one big complex of DNA polymerase is moving. In this case, it's moving along in this direction toward the left side here. Okay, and as it's doing it, it's got a leading strand that's just dropping it right in. Every little bases are being formed. The lagging strand is constantly going into this loop formation back here. Okay, and when that loop formation is where you've got the replication going. Now, it still leaves lots of little primers everywhere that have to be removed, and ligase has to come in to form them. But you can see how this is building this direction. In reality, this DNA polymerase is also building in that direction at the same time. So you have this, you sort of have a constant thing that's working through it. Okay? All right. All right, so that's the basic steps, okay? Again, we've run through these, we've cartooned them. I understand that they're much more complex than this, but we've at least introduced the important things. We've introduced helicase, primase, gyrase, DNA pols. We've introduced lysosine factors, all those things that are important in getting the process started, and then elongation. Okay? All right, now we're not finished. We've got one more set of lectures that we'll need to do. We'll need to talk about finishing all this up and what happens particularly at the ends of the chromosome back down here, what's going on back there, and that's a special event, and that'll be in the next lecture series.